All right, let's go over the muscle types and the sliding filament theory. So as we went over earlier, but just as good review, that there are three main types of muscle tissue, right? So there's skeletal muscle that gives you voluntary control. I mean, my brain can tell me voluntarily to flex, so you decide when to move. They're striated, kind of like when you eat carne asada, right? That striated look, alternating light and dark bands, which we'll go over. They're attached to bone, and a muscle cell, muscle fiber, or a myofiber. That's what it's called. In cardiac, which is your heart, they're striated too, but they're involuntary, meaning you don't have to tell your heart to beat. It just beats on its own, 60 to 100 beats per minute. Sometimes it goes up, sometimes it goes down. You're like, well, if it's going too fast, you're like, slow down. But it's involuntary. You can't tell it to slow down. You got to do some calming techniques, relax, but sometimes it just goes straight up and you get like crazy and you're thinking you're going to have a heart attack. But that's the involuntary component because it's controlled by the autonomic nervous system, which we'll discuss a little bit later. And smooth muscle, which is your GI system, again, involuntary. When I eat something, by the time it goes into the esophagus, I don't have to think about it. I don't have to say, hey, digest. Just does it automatically. That's involuntary. I can't control it. But that's not striated. It's smooth. So if you ever eat menudo, you know that tripe, that's intestine. You can look at that. That's what it looks like. And it has this fusiform cell shape where it's tapered on both ends, kind of like your biceps tendon. All right, so there are over 600 skeletal muscles. There's connective tissue coverings over the muscles. One of the coverings is layers of dense connective tissue called fascia surrounding and separate each muscle. So sometimes when you get those knots in there, that could be fascial pain as well. This connective tissue extends beyond the ends of the muscles and gives rise to tendons. Now remember, tendons connect muscle to bone. So that's what tendons that are fused to the periosteum of the bones. And sometimes muscles are connected to each other by a broad sheet of connective tissue called aponeurosis. So all these connective tissue coverings are found around our muscle. Okay. So if you look at this, here are the layers. Fascia blends with the epimyosin. So if you look here, so here's the deep fascia, here's the tendon. Remember, the tendon gives rise from the fascia. Then it gives rise to the epimyosin right here. The perimyosin extends inward from there. It surrounds bundles of skeletal muscle fibers called fascicles within each muscle. So there's a whole bunch of fascicles here, and the perimyosin is around these fascicles. And then each muscle cell or fiber is covered by a connective tissue layer called the endomyosin, and that's right in here. See that? That's the endomyosin. So here's a fascicle, which is this right here, and the endomyosin is right here. So again, the epimyosin covers the muscle. The perimyosin will cover these fascicles. And then the endomyosin will cover each muscle fiber. And we're going to go down to the cellular level where the myofibril is right here. So again, here's the bone that we studied. Then there's the tendon that connects muscle to bone. Then you have this fascia, and within that fascia then gives rise to the epimyosin. Then the perimyosin is deeper, and then the endomyosin is deeper. Then we get down to the blood, the nerves, and then the cellular level. And we're going to go all the way down here to see how muscles contract using the sliding filament theory. All right, so a muscle fiber, this is just a vocab. Each muscle fiber is a single long cylindrical muscle cell. So a muscle cell, muscle fiber. Fibers respond to stimulation by contracting. Cell membrane of the muscle fiber is known as the sarcolemma. Remember in biology we learned about the plasma membrane or the cell membrane. Well, in the muscle that's called the sarcolemma. Okay, that's like the bouncer of the cell. For the muscles it's called the sarcolemma. And then inside the cell you have this fluid called cytoplasm. But in muscles we call that sarcoplasm. Sarco meaning muscle. So you kind of get prefix and suffix. Sarco is muscle. It contains many mitochondria and nuclei. Remember, mitochondria are the powerhouse of the cells and nuclei are the brain of the cells. Sarcoplasms contain parallel myofibrils, like this is what parallel means, which are active in muscle contraction. You have really 4 proteins that will regulate the sliding filament theory. The 2 contractile proteins are called myosin and actin. The thick filaments in myofibroblast consist of a protein called myosin. So I think thick, sin, myosin, right? It's a sin for thickness. I don't know. But thin filaments in myofibrils are mainly composed of the protein actin. So when you think of thin, whatever way you want to use it so you memorize it for the test, I think that's good. But they also contain these regulatory proteins are called troponin and tropomyosin. So they will regulate the contraction and relaxation, and we'll go a little bit more in detail. But right now I need you to know there's 4 proteins, 2 contractile proteins. The thick myofilament is called the myosin, and the thin myofilament is called the actin. The regulatory proteins are called troponin and tropomyosin. And these filaments produce these bands, so it gives it that striated look. So here's that striation there. Here you go. That looks like a little carne asada, don't you? I must be hungry. It must be dinner time because all I keep saying is tacos and carne asada. Nucleus, there's the muscle fiber. There's the endomyosin. Remember the endomyosin? And there are these striations of light and dark bands. All right, so now more vocab. The sarcomere. So you want to know what a sarcomeres of these myofibrils are made up of many units called sarcomeres joined end to end. Okay, so the functional unit of a muscle is called a sarcomere, and you have a whole bunch of sarcomeres that make up a myofibril. Okay, and one sarcomere is from Z line to Z line. If you know what an accordion is, you know how an accordion goes in and then out. So think of these sarcomeres that will be able to go in and out from Z line to Z line. In the middle we have an M line, and I'll explain to you how it goes in and out like an accordion. Striations consist of an alternating pattern of light, dark, light, dark. The I bands are light bands are made up of actin filaments, which are anchored to the Z line. So here's the Z line, and here are my actin filaments. Okay, and then they make the I band. The A bands are dark bands are made up of overlapping thick and thin filaments. In the center of the A band, you have something called the H zone, which is myosin only. You're like, what? Myosin, actin, thin, A, I, O, M? What's going on here? There's a diagram on the next page that should help you. And the M line, just think M for middle, is the center of the H zone, consists of proteins that hold the myofilaments in place. So the myofilaments, myosin, goes from the M line in the middle, and then the Z line, you have the actin, and then they kind of slide back and forth. So if you look right here, see there's the Z line to Z line, there's the A band, there's the H zone, there's the M line right here, and then they slide in. Look at it, you have hundreds and hundreds of sarcomeres that must contract together to cause me to flex. But from one line to one line here, that's called the sarcomere, and then you have thick filaments, and then these are the thin filaments. Okay? So just to reiterate, a myofibril is made up of a bunch of sarcomeres. One sarcomere is from Z line to Z line, and in between there you have actin and myosin. So here's a sarcomere. Okay, here's the Z line. The right in the middle is called the M line. Okay. Now, beneath the sarcolemma of the muscle fiber lies a network of membranous channels called the sarcoplasmic reticulum. Remember in cells, we used to call that endoplasmic reticulum. You had a rough and a smooth, which is the endoplasmic reticulum of the muscle. The sarcoplasmic reticulum is associated with something called transverse tubules in vaginations of the sarcolemma. Each T-tubule lies within and between 2 cisternae of the sarcoplasmic reticulum. T-tubules are open to the outside of the muscle fiber. The sarcoplasmic reticulum and transverse tubules activate the muscle contraction mechanism when the fiber is stimulated. And I'll talk about sending a nerve impulse, acetylcholine, and then releasing calcium in just a second. Okay, so here's the sarcoplasmic reticulum right here. And then you have these T-tubules, which are right here. Okay, and this will release calcium once we get initiation from the brain. So in other words, the brain has to tell us we need to contract. So we need a brain first. The brain's going to send a signal down the nerves, boom, 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 and it's going to release a neurotransmitter called acetylcholine. And acetylcholine will start the process of making its way through here, and then it will release our buddy calcium. And then I'll take it from here. So let's just review real quick and take it step by step. So let's say we have 4 proteins. Remember, the contractile proteins are myosin thick and actin thin. And then we have regulatory proteins called troponin and tropomyosin. Tropomyosin is going to block the binding site. So we need to move it out of the way. But in order to move it out of the way, we're going to need calcium and troponin, which is coming. All right. So striations called narrow stripes, light and dark, that run perpendicular to the length of the cell. Okay. A band is the dark stripe of thick myofilaments that partly overlap thin. So A bands have thick and thin filaments. The H band, the central region of the A band, that is a little lighter due to lack of thin filaments. And the M line is the midline of the A band and H band that anchors thick myofilaments together. The I band, light strip of thin myofilaments only, and the Z disc or Z line is the middle of the I band. You're like, oh my gosh. I know, for those of you that are visual, you got to see it on the diagram, because all this is just gibberish, right? It's just words. You're like I, H, A, but it's just a way of organizing that sarcomere. And once you look at it, you can kind of see, okay, first start off with Z line to Z line, right? That's the sarcomere. And then in between, you can kind of differentiate the two. But the key is Z line to Z line is one sarcomere. And in the middle, you have the M line. Then go from there. Okay. So here it is. Again, here's a sarcomere, right? If you were to look at this, here's a sarcomere. That's Z line or Z disc to Z disc. These purple ones are the myosin, and those are the thick, and right in the middle you have the M line, the thin filaments, which are actin. Okay. And then so when this Z line or Z disc goes this way, the actin and the myosin pull on each other, and that's what causes the sliding filament. But we need a couple of proteins and we need a little calcium to get that going here. All right, so how do muscles work? Let's put this into perspective here. So it's important to understand the mechanism of muscle contraction and relaxation properly before learning the difference between troponin and tropomyosin. So muscle fibers, we talked about this, are composed of myofibrils. Myofibrils are made up of long proteins arranged in sections called sarcomeres, which are the basic building blocks of striated muscle. Sarcomere has 2 components called thick and thin filaments made up of actin and myosin. Actin thin, myosin thick, which are proteins. Thick and thin filaments of myosin actin are arranged next to each other within the sarcomere. That's what gives it that striated look. The interaction of these 2 proteins within each sarcomere causes the sarcomere to shorten, which in turn causes muscle contraction. During sarcomere contraction, myosin heads in the thick filaments bind with actin in the thin filaments and pull the thin filaments toward the center. So here's the actin, here's the myosin. It's going to come up, it's going to pull it, and it's going to pull it towards the center. That's essentially the sliding filament theory. But the problem is those actin binding sites are blocked by a regulatory protein called tropomyosin. So it blocks it. You're like, you're not getting my actin. So what we need is we need calcium, but calcium's like too weak. So it's going to pick up troponin along the way and say, hey, I need some help moving this bully out of the way. So calcium and troponin come in here and you'll say, hey, get out of the way. And so they will displace tropomyosin, and now the active binding site is open, and then now myosin can hear and slide it through. You're like, what the heck? Really? Yes, that's essentially what all this stuff happens. But the reason you need troponin and tropomyosin is to regulate muscle contraction and relaxation. If you didn't have this regulation, you might stay contracted or you might stay unable to contract. So we need these regulatory proteins of troponin and tropomyosin. So calcium ions are required for the contraction of the sarcomere. When calcium ion concentration rises, the muscle contracts, and when it's low, the muscle relaxes. Troponin and tropomyosin are the 2 regulatory proteins which regulate sarcomere contraction via calcium binding. When calcium ions are present, calcium binds with troponin that I told you, and it removes tropomyosin. It exposes the myosin binding site in actin. When muscle is relaxed, tropomyosin blocks the myosin binding sites in the actin filaments. The key difference between troponin and tropomyosin is that troponin frees the myosin binding sites of the actin filaments, while tropomyosin blocks it. So tropomyosin gets in the way right on actin, and myosin and troponin will move it out of the way. Okay, so picking up where we left off a little bit about the neuromuscular junction. Remember, now the brain needs to say that I need to contract a muscle. It's going to send a signal down the nerves. Okay, and when it sends the signal down the nerves, the skeletal muscle fiber contracts only when stimulated by the motor neurons. Each skeletal muscle fiber is functionally, not physically connected, so there's a little bit of a gap to the axon of the motor neuron, creating a synapse. The neuron communicate with the muscle fiber by way of chemicals called neurotransmitters. Those are like dopamine, serotonin, but in muscles we use a neurotransmitter called acetylcholine. Neuromuscular junction is a synapse between a motor neuron and a muscle fiber that it regulates. So here it is. Here's that motor neuron. It's coming from the brain. It's saying, okay, here it is. And like I said, it's not a direct connection, but what it does is it releases these neurotransmitters, and that neurotransmitter is acetylcholine. And acetylcholine is very important because it's going to start the process of releasing our calcium so it can go find troponin and then displace tropomyosin. Okay. So muscle contraction involves several events that result in the shortening of sarcomeres and the pulling of the muscle against its attachment. The pulling force is exerted by the binding of myosin molecules to actin molecules. The shortening of a muscle fiber results from an increase in the overlap between actin and myosin filaments as they slide past each other. Shortening of muscle fibers results in shortening of the entire muscle, which then pulls on the attached body part to cause movement. You're like, okay. Now, myosin consists of two twisted strands with globular heads projected outward. A group of myosin molecules forms a thick filament, which we talked about. Actin is a globular protein arranged in twisted filaments, a double containing myosin binding site. Binding site is an opening where myosin can come in and bind to it and pull it towards each other. That's what actin has, a binding site. Remember, it's blocked by tropomyosin, and we need troponin to move it out of the way. Right, so those are the regulatory proteins. So troponin and tropomyosin are two regulatory proteins associated with the surface of the actin molecules. Together, these three proteins form the thin filaments. So here it is. Here's the myosin. That's the thick. Here's the thin. Okay, and troponin and tropomyosin is needed to regulate these contractions. So what's the role of actin and myosin? So according to the sliding filament model of muscle contraction, during muscle contraction, a myosin head attaches to the binding site of an actin filament, forming a cross bridge. So here, myosin comes in, it forms a cross bridge. This binding causes the head to bend, pulling on the actin and moving it toward the center of the sarcomere. The head then releases and attaches to the next binding site on the actin, pulling the site toward the center. So it's just boom, 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 and you need ATP along the way. As this occurs again and again, the filaments increase their overlap and the sarcomere shortens from both ends. When many sarcomeres shorten at the same time, the muscle fiber shortens. So energy from the conversion of ATP to ADP is provided to the cross bridges by the enzyme ATPase. ATP breakdown causes the heads to return to the cock position, ready to bind to another binding site. And when you unfortunately pass away, you don't have ATP to break those bonds, and that is what causes rigor mortis. So in order to break those bonds, we need ATP. But as you die, you don't make any more ATP, so you can get rigor mortis for up to 24 or 48 hours after you pass away. And usually those bonds, those bridges will break, but in the first day or two, you can get rigor mortis. Okay, this is probably hard to see on your end, so this is just from the book. That's the sliding filament model, the sarcomere contraction. The stimulus for contraction, again, is acetylcholine, is the neurotransmitter for skeletal muscle fiber contraction at the neuromuscular junction. Acetylcholine is produced in the motor neuron and stored in the synaptic vesicles at the distal end of the neuron. acetylcholine is released into the synaptic cleft in response to an impulse in the motor neuron. It then stimulates the muscle fiber. So if you've heard of Botox, Botox will affect this step right here. So if Botox is hindering the release of acetylcholine, well, then everything afterwards doesn't work. So then you see how Botox works. So if I don't get the acetylcholine to go in and get the calcium to release, then my muscles won't contract. So that's why you have that look for 4 to 6 weeks. Eventually, then the body will get rid of it, and then you're back to normal. And then you got to go get another $1,000 shots. So it's a vicious cycle. But remember, Botox is a neurotoxin. You don't want too much Botox in your body, but the choice is yours. Upon receipt of the muscle impulse, the sarcoplasmic reticulum releases its stored, right? So it needs acetylcholine to release the stored calcium to the cytosol of the muscle fiber. The high concentration of calcium in the sarcoplasmic interacts with the troponin and tropomyosin molecules, which move aside, exposing the myosin binding sites. But calcium, as it makes its way, it's going to pick up the troponin, and the troponin, because calcium and troponin are needed to move tropomyosin out of the way. So calcium and troponin work together to displace tropomyosin. Okay, cross bridges now form and pull the actin filaments using the energy of ATP. This causes the sarcomere to shorten. The contraction continues as long as the nerve impulse continues. After the nerve impulse stops, these events lead to relaxation. So you need an enzyme called acetylcholinesterase. Anything that ends in ACE is usually an enzyme in the motor end plate. Rapidly decomposes the acetylcholine. Calcium is returned to the sarcoplasmic reticulum using ATP as its energy source. Remember, when you die, you don't have this. So if you don't have this, then you cannot, you stay contracted. ATP now binds to the myosin heads, and the linkages between myosin and actin are broken. The actin returns to its original position, and the muscle relaxes. Whoa, I didn't think that it's that involved to just move, right? Yeah, it's a lot of steps, and along those steps, anything can go wrong, right? So I put this steps together. So for contraction, basically an impulse travels down a motor neuron axon. So the brain sends that impulse. The motor neuron releases the neurotransmitter acetylcholine. Acetylcholine binds to these acetylcholine receptors in the muscle fiber membrane. The sarcolemma is stimulated, which is the plasma membrane of the muscle. An impulse travels over the surface of the muscle fiber and deep into the fiber through the transverse tubules. The impulse reaches the sarcoplasmic reticulum, which are those same as the endoplasmic reticulum in cells, and the calcium channels say, "I'm free," and releases. So calcium ions diffuse from the sarcoplasmic reticulum into the cytosol and bind to troponin molecules. So then what they're going to do is they're going to find tropomyosin molecules move, because they displace it and expose specific sites on actin and where myosin heads bind. The cross bridge forms, linking the actin and myosin. Thin filaments are pulled toward the center, so it's pulling the actin, okay? And then the muscle fiber exerts a pulling force on its attachment as contracted. Now remember, hundreds and thousands of these are going on, and this is just one example. You need everything to work together. Well, then how do we relax it? Okay, well, acetylcholinesterase, remember the enzyme, decomposes acetylcholine, and the muscle fiber membrane is no longer stimulated, right? So I needed acetylcholine to get everything going. Well, then if I have too much acetylcholine, it's going to stay contracted. So I need to stop, say acetylcholine, stop. So this enzyme will say, okay, we no longer need it. Calcium, if I don't have acetylcholine, then the calcium ions are actively transported back into the sarcoplasmic reticulum. The ATP breaks cross bridges, linkages between actin and myosin without the breakdown of ATP itself. Breakdown of ATP cocks the myosin heads. Troponin and tropomyosin block the interaction between the myosin and actin, and the muscle fibers remain relaxed, yet ready. If we have acetylcholine again, the cycle happens again and again and again. So in a nutshell, that is the sliding filament theory of muscle contraction. Hope that helps.